Welcome to this last lecture on eukaryotic gene expression, basics and benefits. In this lecture, I am going to introduce to you a very new and exciting area of research in biology and medicine known as metabolic engineering and synthetic biology. Now, what is metabolic engineering? Engineering biosynthetic pathways for low cost production of useful products is a very simple definition of metabolic engineering. That means you alter the metabolic pathways of any organism, and as a result, the culminating out of engineering these metabolic pathways, if you can now synthesize some useful products, it can be a small molecule, or it can be an amino acid, or it can be a flavonoid, it can be a terpene, or it can even be a protein. So by engineering certain specific metabolic pathways, if you can ultimately produce a useful product, that can be known as metabolic engineering. Now, metabolic engineering overcomes the cellular regulation to produce product of our interest or creates a new product that host cells normally do not need to produce. So, there are two important aspects of metabolic engineering. One is usually all these metabolic pathways are very finely regulated, and therefore the intermediates which accumulate in these metabolic pathways are often limited amounts. But by engineering or by tinkering with this regulation of these various metabolic pathways, it is possible to overproduce certain components of these metabolic pathways. We will discuss specific examples later. So, a molecule which is already being produced by a particular organism can be overproduced by engineering some of these metabolic pathways. But in another case, a molecule may not be produced at all by that organism, but by introducing certain new genes, by expressing certain new enzymes, you can now force that organism, or you can force that particular metabolic pathway to produce a compound of your interest. So, either you can overproduce a compound that is already being produced by the organism by tinkering with the metabolic pathways, or you can force the organism to alter certain metabolic pathways so that it can now synthesize a new compound which is not normally synthesized by this organism. So, both examples are possible, and both are being carried out using metabolic engineering. So, let us now try to understand how by introducing certain genes which actually code for enzymes of certain specific metabolic pathways, you can force these organisms. It can be an E. coli, it can be Saccharomyces cerevisiae, or it can be Chinese hamster ovary cells, that is bacterial, yeast or mammalian cell systems, to express certain enzymes or certain molecules of your choice which can be now sold as products. So, by expressing a group of genes, you can now alter metabolic pathways so that you can get a desired product of your interest. That is what is going to be the focus of today's talk. Now, pathway or metabolic engineering for biochemical production is a major research area and is quite distinct from the overproduction of therapeutic proteins. Now, in the last few classes, we have extensively discussed about expression systems, prokaryotic expression systems, eukaryotic expression systems, gene therapy, and so on and so forth. But all through the focus has been on making proteins, how you can overproduce a protein, or how you can knock out the expression of a gene so that a particular protein is not made, or so on and so forth. So, one might ask the question, in what way metabolic engineering is different? Here also ultimately you are making a product, it can be a protein or an enzyme. But the major difference between a metabolic engineering or a pathway engineering is different from a recombinant protein production is that in the case of recombinant protein production, the protein is the final product and machineries already exist in the cells to produce proteins from these genes. You are not tinkering with any pathways to overproduce a protein of your interest. Basically, you are putting a promoter in front of the gene of your choice, and by transcription factors bind to this and your protein of your interest produced. So, you are not tinkering with any pathway. Whereas in the case of a metabolic engineering or a pathway engineering, cells do not typically have the machineries for its production, and multiple genes need to be heterologously expressed to synthesize the enzymes to complete the pathway. So, this is the major difference now. So, for overproducing proteins, you do not have to engineer any pathway because the machinery already exists. You just have to introduce your gene under the control of a particular promoter, and your protein will be made by the organism. But in the case of metabolic organisms, many a times you are not happy with the pathway that exists in the cell. Therefore, you alter the pathway by either tinkering with the enzymes which are existing in the cell, or by introducing new genes so that certain new enzymes can be made so that your desired product can be produced in large amounts. The unique challenges in such engineering efforts include balancing the promoter strengths, enzyme expression, coordinate expression, and interface with the native metabolism. So, why metabolic engineering has become a new discipline or an independent discipline of its own? Because it offers many challenges. Because you have to express a number of genes coding for a number of enzymes of a pathway, and many a time all these enzymes are all under very tight control. They have what is called as a feedback regulation, feedback inhibition. So, if you overproduce or if you underproduce a particular compound or an intermediate in that particular pathway, the reactions do not proceed further. So, a lot of balancing efforts has to be done. The kind of promoter that you use for one particular enzyme of a gene coding for an enzyme of pathway should be different from the promoter that you have to use for another gene coding for another enzyme of a pathway. So, one has to do lot of balancing act in order to make sure that this metabolic pathway functions and ultimately you get the desired product of your interest. That is why metabolic engineering or pathway engineering become an independent discipline of its own and is quite distinct from producing recombinant proteins of your interest. Several biomolecules of therapeutic value are produced by complex metabolic pathways in higher eukaryotes, especially plants. The medicinal plants produce a number of compounds of medicinal value, and they also produce a number of other exotic compounds like flavonoids which give like aromatic compounds, aromatic plants, which are all have a lot of commercial value. So, not only plants are produce products of medical importance, they also produce products of commercial importance which are of non-medical in nature. But the major problem is isolation of these compounds from these natural resources is often cumbersome, inadequate and uneconomical. In most of the times, these compounds which these plants produce are often known as secondary metabolites, and they are often produced in small amounts. So, if you really want these compounds in large amounts, it becomes very difficult to isolate these compounds from a large. We have to really kill a large number of plants and extract these compounds from these natural sources, which becomes uneconomical, and often is the demand of such products is very high. Therefore, what you isolate from the natural source is not adequate and the process is very cumbersome. So, that is why you need to find out alternate ways by which these commercially important compounds or compounds of commercial importance can be generated from alternate sources. So, the challenge in metabolic engineering is, can such molecules which are produced in very small amounts in some of these plants or in further fungal organisms, can they be produced in other prokaryotic or eukaryotic microorganisms by metabolic engineering? That is the challenge that we are addressing now. So, if you have to do this thing, many a times what you have to do is you have to alter the primary metabolism of the host. Many a times when you find that the plant which is synthesizing this molecule probably has a primary metabolic pathway which is common to many organisms, but at some point during this metabolic pathway it branches out, leading to the production of specific secondary metabolites. So, if you want to now produce these secondary metabolites in an organism that normally not produce, you have to now alter this primary metabolism of that host organism such a way that instead of producing a compound which the organism normally produce, you have to divert the pathway in a different direction so that the compound of your interest can be produced. So, you have to alter the primary metabolism of the host, heterologous expression of genes encoding natural product biosynthetic pathway, which is because many of the enzymes which are required for making that particular compound is missing in this host organism. So, you may have to introduce these genes so that those enzymes can be now made, and then they divert the metabolic pathway in a direction that you want. And efficient supply of required biosynthetic intermediates to support the engineering natural product synthesis. Many a times the intermediates which are normally produced in that particular plant may not be produced here. So, often you may have to supplement such intermediates to these organisms so that these intermediates now go and joining those metabolic pathway, and along with the endogenous compounds can now produce the compound of your interest. So, there are a number of challenges in doing metabolic engineering. So, it is not simply taking genes for about 4 or 5 enzymes in a pathway, just put together and you get to your product. That is a lot more complicated than just expressing a bunch of genes. And metabolic engineering is not new. People have been exploiting microorganisms, especially bacteria, yeast and fungi, for producing a number of compounds that have either medical or other non-medical commercial use. For example, one of the very successful compounds which have been produced in large amounts by pathway engineering or metabolic engineering is production of amino acids. Now, annual worldwide production of amino acids is about 400,000 tons. Just imagine the market for these amino acids. Especially two of the amino acids, glutamate and lysine, are one of the most abundantly produced microorganisms using metabolic engineering or pathway engineering. Glutamate, for example, glutamic acid is actually used as preservative in foods called as monosodium glutamate. and it's also used in medicines, many of these amino acids, and many a times these amino acids are starting materials for synthesizing a number of other commercially important compounds. They become the starting material for making number of other compounds. One of the most well characterized amino acid biosynthetic pathways which is commercially exploited is that of Corynebacterium glutamicum. And as early as 1957, this microorganism has been exploited for the overproduction of glutamic acid. There is a very nice review on metabolic engineering of glutamatic production. I will not really go because this does not really, this is primarily based on bacteria. I would like to go to some of the recent developments in the area of metabolic engineering. So, I have just given here, for example, glutamate actually is produced as a byproduct of the Krebs cycle, where alpha ketoglutarate, which is a keto acid, can be converted to glutamate by transamination reactions. So, if you want now the organisms to produce glutamate in large amounts, you can actually introduce certain genes or tinker with certain enzymes of this pathway, so that Krebs cycle is required for energy. So, along with the generation of the energy, you should also be able to produce sufficient amount of glutamate. So, a lot of engineering is done to make sure that while running Krebs cycle, the glutamate that is produced, the organism can also produce large amounts of this glutamate. So, this is an excellent review which actually tells you the historical aspects of how microorganisms were modified and exploited for producing large amounts of glutamic acid. The other example is citric acid. Over 130,000 tons of citric acid is produced worldwide every year, and citric acid is used in foods, beverages, and it's an iron citrate is a source of iron. It's a preservative for stored blood, tablets and ointments, and it is also used in detergents as replacements of polyphosphate. So, it's a microbial fermentation for production of citric acid was developed as early as 1923, and 99% of the world's output is actually produced using microorganism Aspergillus niger, which is a fungus. So, you can see bacteria as well as fungi have been exploited for producing some of these very important molecules like glutamic acid, lysine or citric acid, and so on and so forth. Antibiotics again is a fantastic example. Antibiotics are small molecular compounds that can inhibit or kill microorganisms at very low concentrations, and once you demonstrate that a particular fungus is producing a particular antibiotic or particular microorganism is producing an antibiotic, then you do what is called as a metabolic engineering or pathway engineering to see how you can make that or force that organism to produce large amounts of this antibiotic. Or once you understand how this antibiotic is being produced by this organism, you can actually take these genes or enzymes and express the genes in heterologous organisms, so that this organism can now produce this antibiotic in large amounts. So, a lot of metabolic engineering has been done to overexpress to exploit enzymes of this pathway, leading to production of high levels of antibiotics. And these antibiotics again are products of secondary metabolism, and the antibiotics produced by various bacteria, actinomycetes and fungi like Bacillus, for example, Streptomyces, Penicillium, and so on and so forth. For example, just from Streptomyces alone, you can see so many number of antibiotics are being produced: chloramphenicol, tetracycline, chlorotetracycline, nystatin, erythromycin, clindamycin, streptomycin, and neomycin. So, from different species of Streptomyces, a number of these antibiotics are produced. So, in all these cases, the general principle is that once you know that a particular bacterium or a particular fungus is producing a particular antibiotic, but it not at a concentration that you would like, then you do biochemical engineering or a pathway engineering to see how we can exploit this pathway, so that this particular antibiotic is produced in large amounts. So, enzymes. So, in addition to amino acids, antibiotics, a number of enzymes of commercial importance are also produced using microorganisms. Many enzymes are used in industries, which include amylases, proteases, catalases, isomerases. They all have a lot of commercial value. And enzymes used for analytical purposes such as glucose oxidase, which is extensively used in glucometers for determining your blood glucose. Alcohol dehydrogenase, hexokinase, cholesterol oxidase, all of them again have a lot of commercial importance. So, you need microorganisms which can produce these enzymes in large amounts. Many enzymes are used in medicine such as asparaginase, proteases, lipases, and so on and so forth. So, all these can be overproduced or produced in commercially viable proportions by metabolic engineering. This just tells you how microbial enzymes have tremendous commercial applications. 34% of the microbial enzymes are used as detergents, lipases, proteases to remove the dirt in your clothes. 14% are used in dairy processes for making curd, for making cheese, and so on and so forth. Starch processing, textile industry, beverages and brewing, animal feed, bakeries, a number of other applications. So, producing enzymes by manipulating metabolic pathways so that these enzymes are overproduced in large amounts is one of the major commercial activities, and metabolic engineering has been a very important player in this area. So, it just gives you just a list of the various enzymes and their applications. Bacterial proteases are being used as detergents, asparaginase is an anti-tumor agent, glucoamylases and bacteriomases can be used in fructose syrup production, starch liquefaction, brewing, baking, etc. Glucose isomerase used in sweeteners, rennin is used in cheese manufacture, pectinase is used for clarify fruit juices, lipase is used for cheese production, penicillin acylase, semi-synthetic penicillin, Taq polymerase, of course most of you know, is actually used in polymerase chain reaction. So, you can see producing these enzymes in commercially viable amounts has tremendous applications. Either you can use a recombinant route, or you can actually what is known as a strain improvement or strain modification. You can engineer these strains to overproduce these enzymes so that you can isolate this enzyme either from the native organism or from the recombinant form. So, let us now start, having looked at the importance of metabolic engineering and understood what kind of molecules are being produced by metabolic engineering for a number of years, right from 1930s. Let us now try to understand how do you go about doing metabolic engineering. Now, suppose you want to produce a molecule of your interest by metabolic engineering in prokaryotes or eukaryotes, how do you go about? Now, you can do metabolic engineering in prokaryotes as well as eukaryotes. In fact, the Escherichia coli, Bacillus subtilis, these organisms have been extensively exploited for metabolic engineering of number of compounds of commercial importance. The ease or the advantage of using prokaryotic hosts for metabolic engineering is that prokaryotes have evolved operons, which we have studied in the first lecture of this series. For prokaryotes have evolved operons for the economical co-expression of several genes required to achieve a desired metabolic state. So, we already know, for example, you have a lac operon, trip operon, so on and so forth. So, the prokaryotes are already tuned to expressing all the genes required which code for an enzymes of a particular metabolic pathway. So, if you want now overexpress enzymes of a particular metabolic pathway, all that you have to do, you have to take the genes code of this particular pathway, put them under operon and see whether you can get one single polycystonic messenger RNA and all the enzymes can be made in one shot. So, because genes coding for metabolic enzymes are grouped into operons, it is often possible to engineer metabolic pathways much more easily in prokaryotes, because by manipulating a single operon you can produce all the enzymes of a particular pathway. So, these operons, as we all know, typically consist of a promoter which drives the expression of a single multicystonic message, which we studied long back about how lac operon, trip operon and so on, they all work. However, when you come to eukaryotes, there are no operon concept in the eukaryotes. Majority of the genes are organized in monocystonic regulons scattered across the mammalian chromosome. So, if you have 5 enzymes that are required in a particular metabolic pathway, each of these enzymes, the gene coding for each of the enzymes may be present in a different chromosome. So, unlike in prokaryotes where all of them are present in a cis arrangement under the coordinate regulation of a single operon. So, one of the strategies for expressing multiple genes of a pathway in mammalian cells is to assemble a multicystonic or eukaryotic operon by cloning several internal ribosome entry site containing translation units in consecutive orientation under the control of a single eukaryotic promoter. Now, when we talked about eukaryotic expression plasmid, we discussed extensively about the use of internal ribosome entry sites, how it promotes cap independent translation initiation, and how by using internal ribosome entry sites you can express multiple genes under a single promoter. So, by creating vectors containing such internal ribosome entry sites, one can often produce up to 3 to 6 proteins in a single plasmid or in a single regulon. The terminal cystone usually contains a single polyrelation site for proper processing of this multicystonic messenger RNA. So, by using this concept, by generating this kind of artificial constructs containing a promoter, gene 1 followed by internal ribosome entry site, gene 2, internal ribosome entry site, gene 3 and so on and so forth, and the final cassette ultimately containing polyrelation signal, one can produce a multicystonic message in eukaryotic messenger RNA in eukaryotic cells, which ultimately gets translated. The first gene gets RNA gets translated in a cap dependent fashion, whereas the subsequent RNAs in this multicystonic message get translated in a cap independent fashion, and you can get all the proteins or enzymes of that particular pathway. This is one strategy. Now, one of the what kind of compound that you would like to produce by metabolic engineering? The compound that usually are produced in large amounts are those which are actually produced normally in plants. The plants produce such useful compounds both as a result of primary metabolism or as a result of secondary metabolism. Now, what are the plant primary metabolites? These are organic compounds produced by the plants. They have metabolic functions essential for the plant growth and development. They are produced almost in every plant because these are primary metabolic pathways, so they must be present in all the pathways, and these include metabolic pathways involved in carbohydrate metabolism, amino acid metabolism, nucleotides, fatty acids, steroids and lipids. These pathways are common almost to all plants or even for animal kingdom. However, the secondary metabolites are organic compounds. They are also produced in a number of plants, but usually they do not have apparent functions involved in plant growth and development. They are produced in different plant families, in specific groups of plant families or in specific tissues of a plant, or either throughout the development or in specific stages of the development. Such secondary metabolites include terpenoids, special nitrogen metabolites such as amines, cyanogenic glycosides, glucosylulates, alkaloids, phenolics and so on and so forth. So you can see the secondary metabolites, which are actually of great commercial importance, are not present in all the plants, and they usually do not have a primary role or in primary metabolic pathways, and they are only produced in a handful of plants, and even a plant they may produce only in certain tissues or certain cells. Some of the examples of such secondary metabolites which have extensive commercial importance are, for example, alpha linolenic acid, which is a PUFA or a polyunsaturated fatty acid, which is actually present in walnut and flax seeds. Polyunsaturated fatty acids play a very important role for human nutrition, especially for the normal development of a child. You require these polyunsaturated fatty acids. Flavonoids, terpenes like such as paclitaxel, artemisinin is a very potent antimalarial compound. Paclitaxel is a precursor for Taxol, which is a very important anti-cancer compound. You have flavonoids which are used in aroma industry. These are all secondary metabolites produced by select group of plants, but not produced by all the plants. Caffeine, for example, it's an alkaloid. Again, nicotine produced by Nicotiana tabacum. Allicin, which is a non-protein amino acid. Caspicin produced by capsicum. These are all examples of secondary metabolites which have tremendous commercial importance, but is not produced by all the plants. Just as the plants produce many of the secondary metabolites which have important commercial value, many fungi also produce a number of secondary metabolites like non-ribosomal peptides which have a lot of medical importance, beta-lactam antibiotics, cyclosporin, a potent immunosuppressant, echinocandin, an antifungal drug, polyketides, lovastatin, a potent cholesterol lowering drug, aflatoxin, ergotamine used in migraine treatment, and so on and so forth. So not only plants, even the fungi have the secondary metabolic pathways, and these secondary metabolites which are produced by plants or fungi have enormous commercial importance. Now, as I said, once we understand what kind of compounds are being produced by the primary metabolism and secondary metabolism, and these secondary metabolites being produced by plants or fungi, the question comes, why do you have to do metabolic engineering? As I explained above, although these plants or fungi are producing these compounds are usually not produced in a commercially viable propositions. So if you want to make kilogram amounts or tons of these compounds, these natural sources are inadequate. So unless you do metabolic engineering, unless you make this organism to overproduce these molecules, or unless you transfer these secondary metabolic pathways to a heterologous organism so that this organism now can produce these secondary metabolites in not large amounts, these will not be a commercially viable proposition. That is where the metabolic engineering becomes very, very important. And as I said, many of these compounds are produced by complex metabolic pathways involving several enzymes, and therefore if you have to do metabolic engineering, you have to do what is called as a multi-gene expression. Just by expressing one gene, you cannot get, say, secondary metabolite of your interest. Often at least half a dozen to 1 dozen enzymes have to be overexpressed or has to be engineered so that you can alter the metabolic pathways and overproduce the compound of your interest. So I will give one particular example, Taxol. Now Taxol is a widely used anti-cancer drug that is usually isolated from the bark of Pacific yew tree called Taxus brevifolia. Now let us see the logistics here. Now one has to sacrifice 2 to 4 fully grown trees to get sufficient Taxol for the treatment of just one patient. Just imagine, although Taxol is an anti-cancer compound, if you want to treat one patient with Taxol, you have to cut 2 to 4 fully grown trees and then isolate this compound from the bark. In fact, during the early stages of its clinical use, 130 kg of Taxol was extracted from 1000 tons of the bark, which required cutting down of more than 500,000 mature trees. So you can see what an ecological disaster it is, because you cannot just, now suppose we have to meet the global needs, it is impossible for you to make Taxol by going on cutting trees like this and isolating this compound from the bark. Whereas at present nobody is cutting these trees to make Taxol anymore. At present, Taxol is being produced either through chemical conversion of a related molecule derived from the needles of a more prevalent European view, that is Taxus baccata, or by plant tissue culture. So there are alternate methods now to produce this Taxol in large amounts, and you don't have to go and cut these trees in large scale. I want now just spend some time to explain a very recent paper that actually came in Science just last month in October 2010 about how you can engineering this Taxol biosynthetic pathway to produce a Taxol precursor using E. coli. So initially, once Taxol was discovered as an anti-cancer compound, it was being isolated from the bark of these huge trees, so I have to cut down large number of trees, but then people went and then started producing other fungi, or they started producing by plant tissue culture. But now people are trying to see whether microorganisms can produce Taxol. Can you engineer this Taxol biosynthetic pathway into microorganisms like E. coli or yeast, so that these microorganisms can now produce Taxol in large amounts? And this is a recent paper, so I will just spend some time explaining what is the highlights of this particular paper. Now, as I said, if we have to understand how metabolic engineering can be done for Taxol, we need to very briefly understand how Taxol is biosynthesized. Now, Taxol belongs to a large family of compounds known as terpenes, which include artemisinin, which is a very potent antimalarial, carvone, a flavoring agent, pinene, which is the main component of terpentine. So all these very commercially important terpenes are all produced from a common metabolic pathway. Now the carbon skeletons of all these terpene molecules are made from a compound called isopentenyl pyrophosphate and dimethyl allyl pyrophosphate, IPP and DMAPP. So these are the common precursor for the synthesis of not only Taxol, but also many other commercially important compounds like artemisinin, carvone, or pinene, and so on and so forth. Each terpene up to a certain steps, the pathway is common for the production of all these terpenes. After then they branch out, and these different branches now produce different kind of these terpenes. Each terpene skeleton is then modified by a series of enzymatic reactions, ultimately leading to synthesis of the final natural product. Now the conversion of this IPP and DMAPP into Taxol is a very complex process that is present in the cells of only that particular tree, that is the Pacific yew bark. Okay, that is why only this particular tree produces Taxol. So now let us understand and see how we can now engineering this pathway of Taxol production into a non-producing organism like E. coli or yeast. So to put in a nutshell, glucose, as you know, is the carbon source for all organisms, and this glucose during normal metabolism is converted into this IPP or DMAP in most of the organisms. There are two pathways by which this glucose is converted to IPP or DMAPP. One is called as a mevalonate pathway, and another is called as a non-mevalonate pathway. Now what I am going to tell you in the next few minutes is to see how we can engineer or how you can tinker with both this mevalonate pathway and non-mevalonate pathway, so that you can now make microorganisms produce either Taxol or artemisinin in large amounts. That is what is it. So let us now spend some time to understand how this particular group, Ajit Kumar et al., in the just about 1 month back, have engineered an E. coli strain so that it can now start producing a Taxol precursor using this particular pathway. So what exactly these people have done? Let us now understand what the reality and what these people have achieved. Now the precursor for Taxol is called taxadiene. Usually in E. coli, so far the maximum that one could achieve by metabolic engineering is about 8 mg per liter per hour. That is the level that they could produce. In this particular paper, these people have been able to demonstrate that by doing metabolic engineering, they can actually produce up to 60 mg per liter of this Taxol. So several fold increase has been done. Well, let us now just understand how this was done. This was done by a number of series of very innovative steps. First, what they did is that improvement in the rate of IPP and DMAP synthesis by careful optimization of the absolute and relative levels of these 4 key enzymes in E. coli. As I told you, when we do this metabolic engineering or when we do this pathway engineering, one has to be extremely careful about the levels at which various intermediates in pathway is accumulating. So, if you randomly overexpress all the enzymes of this pathway, ultimately you will not get the result, because you require what is called as a metabolic flux. Many of these intermediates have to be retained in specific ratios in order for this pathway to drive in a particular direction. So, what these people actually did is that they actually carefully optimized by using appropriate promoters. They made sure 4 of the key enzymes in this pathway is expressed in appropriate levels that will drive the reaction in a favorable direction. The intracellular activity of 2 plant enzymes called GGGP synthase and taxidine disynthase were enhanced substantially by codon optimization and deletion of an N-terminal plasmid transit peptide. So, it's typical genetic engineering. So, they optimize these codons of this so that you use only codons which are very highly expressed in E. coli, so that E. coli optimized codons were used for making artificial genes so that they can be expressed in high amounts. The upstream segments to the IPP, DMAP and downstream segments to taxidine were balanced by varying the promoter strength, plasmid copy number and genotype. As I said, you cannot put the same promoter and same regulatory elements for all the genes of the pathway. Depending upon the need, if you know, for example, enzyme 1 is required high levels and enzyme 2 is required lower levels, you need to put the appropriate promoter so that this kind of a ratio is maintained. So, all these things were taken care. And of course, the last thing is engineering a chimeric cytochrome P450 reductase enzyme harboring both oxygenase and reductase activity without membrane anchor. Now, usually these cytochrome P450 reductases, they are all membrane proteins. Without a membrane anchor they will not function, but they have been able to engineer a cytochrome P450 reductase enzyme which has both oxygenase and reductase activity even without such membrane anchor. So, you can see a number of modifications were done to their expression system, and by doing all these things they were able to engineer an E. coli which now produces very high levels of a precursor for Taxol, and this precursor then chemically can be converted into Taxol. So, many of these things are called the semi-synthesis of Taxol. That means you have not engineered the entire biosynthetic pathway, but up to a point you produce an organism, and once the precursor comes, and by just another 2 or 3 steps you can make the final product. So, it is called a semi-synthetic method. So, this is how this recent study has actually shown that by taking care of these parameters which I just explained, one can now make a bacterium like E. coli to produce a Taxol precursor in very high amounts, and this precursor can then be converted into Taxol by chemical means. Let us now spend some time to understand another very important paper that again came just last year about how yeast cells were actually engineered to produce artemisinin, which again is a very potent antimalarial and is in a very high demand. In fact, is one of the most potent antimalarials known today. So, how did people engineer semi-synthetic route for artemisinin production? That is what we will go and discuss in the next couple of minutes. The 2 papers which created a lot of noise, or which attracted a lot of attention among the researchers, is the one that published in 2006 in Nature, entitled Production of Antimalarial Drug Precursor Artemisinic Acid in Engineered Yeast. So, yeast cells were actually engineered to produce artemisinic acid in large amounts, and this artemisinic acid by another 2 or 3 steps can be converted to the final artemisinin by chemical means. And there is a very nice news and views in Nature on that particular issue saying malaria breakthrough raises spectre of drug resistance. What they actually did is that there is a primary metabolic pathway in almost all organisms in Saccharomyces called as mevalonate pathway. This actually produces mevalonic acid and is actually involved in cholesterol biosynthesis, and this pathway is common for all organisms. So, what these people actually did is that they engineered this mevalonate pathway in the Saccharomyces cerevisiae in such a way that after a certain step this mevalonate pathway branches off, and instead of producing the normal end products which are seen in the Saccharomyces cerevisiae, it now produced artemisinic acid. So, what did they do? I am just going to highlight, I am not going to give all the details because it is a pretty complex paper and one has to spend a lot of time to understand how exactly they did, but the purpose is not to confuse you, just to give you the highlights so that you can appreciate how metabolic engineering is done. So, what these people did? First they did is they engineered the farnesyl phosphate biosynthetic pathway to increase farnesyl phosphate pyrophosphate production. This is one of the very important intermediates in the synthesis of this artemisinin. Then they introduced what is called an amorphodine synthase gene from Artemisia annua. Now, this is a plant from which the artemisinin is normally produced. It is actually a Chinese herb which is grown only in a specific geographical region, and this was first discovered as an antimalarial by Chinese and is present in this Chinese herb. So, they took this gene from this Artemisia annua, which is a gene called amorphodine synthase gene, into the high FPP producer, so that this FPP can now be converted to amorphodine. So, the enzyme amorphodine synthase, which is not normally present in Saccharomyces, but which is present only in Artemisia annua, has been now engineered into the Saccharomyces cerevisiae, so that when you now express this enzyme, FPP now gets converted to amorphodine. Next, they cloned a novel cytochrome P450 that performs a 3-step oxidation of the amorphodine into artemisinic acid from annua and expressing it in an amorphodine producer. So, they took another cytochrome P450 enzyme from this Artemisia annua, so that you can now convert in a series of steps this amorphodine can be now converted into artemisinic acid. So, you can see first they produced a Saccharomycin which can produce large amounts of this farnesyl pyrophosphate. Then they took a gene which is normally present in the Artemisia annua and put it in the Saccharomyces cerevisiae, so that this FPP can get converted into amorphodine. Then they put another enzyme so that this amorphodine can get converted into artemisinic acid. So, this is basically the juggler they did to make this artemisinin, known as the semi-synthetic biosynthetic route for artemisinin production. This is the normal pathway. Acetyl-CoA up to mevalonate and farnesyl phosphate is the primary metabolic pathway which is already present in Saccharomyces cerevisiae. What they did is they tinkered here a little bit so that this farnesyl phosphate can be produced in large amounts. Then they introduced 2 enzymes from Artemisia annua, so that this farnesyl phosphate can now be converted to artemisinic acid, and the green arrows indicates the biological synthesis, and the red arrows indicates the chemical synthesis. So, once you have artemisinic acid produced in large amounts in Saccharomyces cerevisiae, you isolate it, and by about 2 or 3 steps in vitro by using chemistry, you can convert into final artemisinin. In fact, in 2008 these people have worked again on these organisms, and then finally now in this particular paper they actually show that you can even produce up to 1 gram of artemisinin per liter by doing this kind of a bioengineering business. So, to put it in a nutshell, what metabolic engineering basically is, it involves isolating genes from the higher eukaryotes. It can be a plant or it can be a fungus, and then express them using appropriate promoters in your host organism of your choice. It can be an E. coli or it can be an yeast. But since plant genes are not very efficiently expressed in E. coli, you have to do what is called as a codon optimization. So, you synthesize genes which contain codons that are very highly used in that particular organism. For example, want to express in E. coli, you optimize the codon for high level expression in E. coli, or you use those codons which are highly expressed in yeast if you want to express in yeast in large amounts. So, synthetic genes with codons optimized for high level expression in E. coli has to be used, and then you assemble the genes into synthetic operands if you want engineering a pathway in E. coli, generate strains in which specific genes of the endogenous pathway are knocked out, and prevent the accumulation of intermediates which often inhibit growth. So, a series of modifications has to be done so that this strain, this bacterium or yeast now produces the compound of your choice. I want to just spend 2 minutes to explain this particular aspect, which is very, very important. In fact, is one of the major bottlenecks when you do metabolic engineering, that is how to prevent accumulation of intermediates which are often growth inhibitory. Now, as I said, many of these metabolites many of the intermediates which are synthesized during these metabolic pathways, they have to be present in appropriate amounts. If you either produce if these intermediates accumulate either in low amounts or high amounts, it is detrimental to the organisms. The organism may not grow at all. So, you cannot produce high amounts of your end product if the organism does not grow. So, you have to do a lot of what is called as the balance the flux. Balancing the flux of the synthetic pathway has to be very, very important, and if you do not balance this flux, these intermediates will accumulate in inappropriate amounts, and then your engineering will not be successful. So, one way to balance this flux is through transcription optimization of various genes of the pathway. This is what I told you. You cannot use the same promoter for expressing all the genes of the enzymes of this pathway. If a particular intermediate has to be present in large amounts, you have to use a strong promoter. If a particular enzyme has to be expressed in low amount, you have to use a low promoter. All this balancing has to be done so that this flux is maintained. Or by using a library of promoters to various enzymes in an isoprenoid production pathway, it is possible to engineer a flux balanced pathway with improved yield and reduced metabolic purulence. So, this operand often should contain a combination of promoters and enhancers so that this flux is maintained. In fact, well-characterized families of transcription regulators have emerged as very powerful tools in metabolic engineering as they allow rational coordination and control of this multiple gene expression. So, unless you understand how these various promoters work and how you can manipulate these various promoters, it is not possible to metabolic engineering, and that is why understanding this regulation of gene expression very important. You need to have a bunch of promoters in your hands. Some of them are expressed high levels, some of them are expressed in low levels, and by putting them all together one can do this pathway engineering and prevent this balancing, and can balance the flux so that these intermediates do not accumulate in large amounts. I think can just recommending this particular paper, balancing a heterologous malonate pathway for improved isoprenoid production E. coli, came in 2010. Kiesling is one of the major authorities in metabolic engineering. It's a very nice review. One can read and then try to understand how this is done. The other example, which I will not discuss in detail, is about producing ethanol in microorganisms. As you know, today everybody is talking about non-conventional energy. People are trying to see, can you find alternatives for petroleum? So people are now trying to see, can you now produce biodiesel? Can you produce ethanol, which all can have when be used as biofuels? Can you produce from corn? Can you produce from microorganisms like bacteria? Can you produce from fungi? So one of the very attractive choices for metabolic engineering of producing ethanol is yeast. Why? Because yeast is a natural producer of ethanol, but it is unable to break down cellulose and hemicellulose, major components of the biomass. As you can see, we have large number of plants, and so you have a large amount of waste material which are all derived from cellulose and hemicellulose, which is actually called biomass. But unfortunately, if we can now somehow convert this cellulose by enzymatic gradation pathway ultimately to ethanol, you have solved the energy problem of the world. So one of the major focus that is happening in the area of biotechnology is to see how you can degrade this biomass, which is present in large amounts in the form of cellulose and hemicellulose, and how you can break down this glucose from the cellulose and hemicellulose and ultimately convert into ethanol. So although yeast has been known being used for conversion of ethanol, yeast can only produce ethanol from glucose, but it doesn't have the machinery to break down the cellulose and hemicellulose. So engineering yeast to break down cellulose and hemicellulose and utilize all the sugars from their breakdown is currently a major research area in the area of bioenergy. In order to endow yeast with these above functions, multiple enzymes and pathways needs to be inserted and optimized. It's not just easy. For example, I have Saccharomyces cerevisiae, which can only convert glucose to ethanol. But you want cellulose and hemicellulose to be broken down to glucose and then the glucose to be converted to ethanol. So you need to now introduce those enzymes which can break down cellulose and hemicellulose, can be converted to glucose, so that it can now then convert to ethanol. For example, to break down cellulose, you need to introduce all these genes into Saccharomyces cerevisiae. Similarly, if you want to break down hemicellulose to xylose or arabinose, at least 4 classes of enzymes are required: xylanase, xylosidase, glucuronidase, arabinose. So you can see, unless you engineer this pathway, unless you introduce this cellulose or hemicellulose degradation pathway into yeast like Saccharomyces cerevisiae, you cannot break down cellulose or hemicellulose and convert them into ethanol. So a major area of research in the area of bioenergy is focused on how to degrade this abundant biological waste. So much of biomass is generated every year by the decay of this plant material, and if you can somehow convert all this bio waste into ethanol, the energy problem can be solved. So I will not go into the details of this. I suggest that you read this very nice review, Ethanol Production from Xylose and Engineered Saccharomyces cerevisiae Strains, Current State and Perspectives, which actually gives you a very modern perspective of what kind of efforts are actually going on to do metabolic engineering so that yeast like Saccharomyces can be engineered to break down cellulose and hemicellulose so that you can generate ethanol from these sources. Okay. So having emphasized the importance of pathway engineering for producing useful molecules like Taxol, artemisinin, ethanol, and so on and so forth, I want to now spend maybe about another 5-10 minutes in explaining another very important area of metabolic engineering, namely engineering glycosylation pathways for the production of humanized glycoproteins. Another very fascinating area of research that's happening in the area of metabolic engineering. Now let's now understand why you have to do this. What we are now going to discuss is how you can develop glyco-engineered yeast strains. Now let's understand the importance of glycoproteins. Now glycoproteins, that is, these are proteins in which carbohydrate moieties are attached. Glycoproteins comprise about 70% of all approved therapeutic proteins. You take many proteins like tissue plasminogen activator, many of these growth factors, many of the cytokines, they are all glycoproteins. And it is estimated that approximately 30% of the new drugs that are likely to be licensed during the next decade will all be based on antibodies. Again, antibodies are glycoproteins. Glycosylation plays a crucial role in determining biological activity, half-life, distribution of these proteins. So, if you have a properly glycosylated protein, you have hit the right thing, because that is what you want. Human IgG, if you take for example, it is a glycoprotein, and serum IgG consists of multiple glycoforms owing to the differential glycosylation of the Fc region of the antibody. While glycosylation does not affect the antigen binding of the antibody molecule, it has profound effect on the biological mechanisms that are activated by immune complexes formed, as well as the solubility and stability of the antibodies. So, although the glycosylation is not important for the antigen antibody resection, it has very other important functions. Now, for producing this kind of a glycoproteins, including antibodies, the most preferred expression system has been the mammalian expression system. Mammalian cell culture, especially the Chinese hamster ovary or the CHO cells, emerged as the expression host of choice for production of therapeutic glycoproteins. To date, mammalian cell culture has been the only process to yield human-like glycoproteins, but in some cases human proteins expressed in mammalian cell lines also poses a problem, because the glycosylation pattern is different in different species. It is even within mammals, it is different from humans to other mammals. For example, glycoproteins produced from human cells usually contain a mixture of both 2,6 and 2,3 sialic acid, whereas the sialic acid linkage in CHO cells is exclusively of 2,3 type. So, you can see the way these sialic acid residues are added to the protein moiety is different in CHO cells versus in humans. So, to overcome these problems, CHO cells have been engineered to express this 2,6-sialyl transferase gene. So, if you now introduce this 2,6-sialyl transferase gene, so the CHO cells also now can do this, so that you can get a protein which has both 2,3 as well as 2,6 sialic acid linkages. So, expressing these glycosyl transferases, that is the enzymes which add this carbohydrate moiety to proteins, are called as glycosyl transferases. Expression of these glycosyl transferases is now has become one of the most important emerging areas of biotechnology, and this is actually known as the Glyco Engineering. But one of the problems in producing these kind of a glycoproteins in mammalian systems is that, as I told you in long back, mammalian expert systems are very expensive. Maintaining CHO cells and producing proteins in CHO cells is very expensive. So, question is, can we produce these large proteins in a yeast? Can you do this kind of a human glycosylation of proteins in yeast system, so that glycoproteins can be now produced in an economical manner in yeast cells? That is the challenge. Protein glycosylation in yeast is also very different. Although yeast does glycosylation, the glycosylation that happens in yeast is very different from mammalian cells. So, when mammalian glycoproteins are produced in yeast, high mannose type N glycans are attached to the recombinant glycoproteins, which can be cleared rapidly from the human bloodstream due to interaction of the human mannose receptors, and they can cause immunogenic reactions in humans. So, to overcome these problems, it is necessary to re-engineer the glycosylation pathway in the yeast, so that it now produces glycosylation pattern which is very similar to that in humans. So, how do you do that? One approach is to delete certain yeast specific genes involved in hypermannosialization, that is, delete certain genes coding for this mannose transferases, and then introduce genes of other enzymes which are normally present in the humans. So, I am going to give one example to illustrate this point. Here is an example. As you know, glycosylation takes place in two cellular compartments. One is endoplasmic reticulum, another is Golgi. Now, when you look at the glycoproteins in either humans or Pichia pastoris, which is the yeast, in the endoplasmic reticulum, the glycosylation pattern of the protein is more or less the same. The red here is the protein, and this is the carbohydrate point. The different color shows different carbohydrate moieties. So, in the endoplasmic reticulum, where N glycosylation takes place, the pattern is very similar to that of humans and pastoris. But after that, when the protein enters the Golgi compartment, the final product that comes out of the Golgi in human, you can see the glycosylation is quite different from that of the Pichia pastoris. So, although the glycosylation pattern is very similar in endoplasmic reticulum, after Golgi, the product that comes out in Pichia is quite different from that in the human. So, the challenge now is, the glycoprotein that is produced in Pichia pastoris Golgi should now look like the protein that is produced in the human Golgi. How do you do that? This is what you do. You delete two genes called as mannosidase 2 and 1,6 mannosyl transferase in this Pichia pastoris. So, in the endoplasmic reticulum, you now end up with a glycosylation pattern like this rather than like this, and you can see the glycosylation pattern is different here. Then you express a series of glycosyl transferases which are normally will do human type of glycosylation, so that you now ultimately end up with a glycoprotein. You can see now this looks very similar to that made in the Golgi of human beings and not the same as that is being made in the Pichia pastoris. So by removing certain Pichia pastoris glycotransferases and by introducing certain new glycotransferases, you have now ended up with a glycoprotein which now looks very similar to that made in the human Golgi rather than the yeast Golgi. I suggest you read this very nice article, Glycosylation Engineering in Yeast, the advent of fully humanized yeast in current opinion biotechnology, which gives the entire details of how this metabolic engineering has been done. There is actually a company called Glyco which has pioneered this kind of a engineering yeast cells for producing humanized glycosylated proteins, and if you go to this company website, you can understand much more about how these humanized glycoproteins can be made in yeast cells. I think I will skip some of these things. I would like to now spend about 5 minutes expressing another fascinating area called synthetic biology, which is actually an extension of metabolic engineering. What is synthetic biology? The goal of synthetic biology is to develop designer organisms for the sole purpose of serving people. To accomplish this goal, scientists first need to understand what is life. To address this problem, scientists have taken two main approaches. In one approach, the organism is stripped to its bare minimum genome in an effort to understand what is required to create self-replicating organisms. What is the minimal number of genes that is required for an organism to self-replicate? That is the first challenge. The second, once you understand this, can you now actually synthesize these genes and make an artificial organism which contains genes which have been artificially synthesized? And if you now put these genes into a shell of living organisms, you have created artificial life. This is the challenge of synthetic biology. Can you create artificial life? Now, in fact, many things have been achieved. So, I am going to just take 5 minutes to explain what has been achieved in the area of synthetic biology. There is one thing that started one of the first challenges of the synthetic biology is what is called as a minimum genome project. Mycobacterium genitalium is an obligate intracellular parasite whose genome consists of about only 482 genes. Now, human genome consists 30,000 genes. So, this is a much smaller organism. And this is arranged in just one chromosome, and this is the smallest genome of any known natural organism. So, they took this organism, which is an independent living, which is a self-replicating organism, and asked the question, among these 482 genes, how many genes are actually required? What is the minimum amount of genes required for this organism to self-replicate? And they found out a minimal set of 382 genes is required that can sustain life. To sustain life, you require just 382 genes. That is what came out of this. So, this is known as the minimal genome. So, this minimal genome containing 382 genes, if you put it in an organism, it can replicate itself. Then the challenge is, can you synthesize chromosomal DNA sequences consisting of 382 genes, and if now put them into another mycobacterium species which does not have its own DNA, now this artificial DNA can now replicate, and you have created basically artificial life. So, can you now generate what is called a mycobacterium laboratorium, which now replicates with the man-made DNA? That has been the excitement. So, to achieve this, you cannot go and actually synthesize 382 genes straight away because it is too large amount of DNA. So, in December 2003, Craig Venter, one of the pioneers in the area of synthetic biology, actually synthesized about a 5000 base genome of a bacteriophage called 5X174 in about 2 weeks time. So, you can actually synthesize about 5000 base pairs of DNA in about 2 weeks time. But the mycobacterium genome is much bigger. So, it took at least another 5 years before they can synthesize the entire 582,000 base pair gene chromosome of Mycobacterium genitalium, and then they put this into another Mycobacterium species and created a new bacterium which has a man-made genetic material, and this was named as Mycoplasma genitalium JCVI. The synthetic biology is extensively being done in this institute called Craig Venter Institute in San Diego and California, and if you actually go and then click on this website, it gives you a very nice video and extensive literature on the synthesis of the first self-replicating synthetic bacterial cells, how they went about and how they created artificial life. So, this is basically in a nutshell this is what they did. They constructed a series of DNA fragments of 5 to 6 kb length, about 100 such pieces, and then in vitro by recombination they assembled all of them, and then 25 such overlapping DNA fragments of about 17 to 35 kilobase each were then put into yeast cells, and once you put these pieces of DNA in yeast cells by in vivo homologous recombination, they got assembled into a huge chromosome and it started replicating, and they have basically created what is called as an artificial life. and was estimated that this synthetic genome costed about $40 million US and took about 20 people, and they worked for about 10 years to create this artificial life. This is the smallest genome, a microorganism containing the smallest artificial genome. So what I would like to now say is that metabolic engineering and synthetic biology are very exciting areas of research. Fantastic things are being out, and we are almost very close to creating what is called as an artificial life. And I can tell you, this lecture series on eukaryotic gene expression, in all these lectures, in all these topics, I have made sure I have stressed how important is understanding gene expression. Without understanding gene expression, without understanding how gene expression is regulated, you cannot achieve any of these things. So understanding gene expression is crucial for the development of transcriptional gene circuits by chemically interconnecting multiple promoters, repressors, activators for pathway engineering, as well as multi-gene expression pathways in metabolic engineering as well as for synthetic biology. So I have given a series of references here one can go through. There is a tremendous excitement in the area of metabolic engineering as well as synthetic biology, and we are not very far away of creating designer organisms, designer microorganisms that will actually make any compound of it. You can engineer any metabolic pathway into a microorganism, you can produce any compound of your interest, and you can actually make what is called as the designer organisms that will produce what you want by doing all this synthetic biology. Again, a series of references one can go through, and I would like to conclude that this is the last lecture in this 40 lecture series, and I hope that you enjoyed this 40 lecture series on eukaryotic gene expression basics and benefits. We began by discussing very basic aspects of gene regulation, and I have taken you through a journey of about 40 lectures of explaining not only many basic aspects of gene regulation, but wherever possible I have explained to you what are the applications that is coming out, and that hopefully will come out. A lot more exciting applications are being awaited in the near future, and this 21st century actually belongs to biology and biotechnology. A lot of exciting things are happening in the area of eukaryotic gene expression, and many more interesting things are likely to come out of eukaryotic gene expression. And if you have any questions or any clarifications, you can email to me at this email address, pnr@biochem.iac.ernet.in. Thank you and goodbye.